So today our biology is at what we call protein synthesis. Remember, as protein synthesis is such a process which is very, very vital in biology. Remember, we eat food. The food must be broken down by enzymes. Remember, we need it to form structures like keratin. Protein is enabling us to form structures. For instance, the air, the nerves. So remember, we have to carry oxygen in the body. We are using a protein, hemoglobin. Now, the oral manufacture and the oral manufacturing process of such proteins, which are very, very vital in biology. Guys, this is what we are going to call protein synthesis. So in simple terms, we can biologically say protein synthesis is a process of forming particular proteins like enzymes, keratin, hemoglobin, and so on. Now, to understand the process of protein synthesis properly, members, we have to understand where the process occurs. Mainly, if you go back to your cell biology or cytology, you discovered, or the teacher told you, that protein synthesis mainly occurs in what we call the ribosomes. We are going to see ribosomes or smaller unit and then a big unit of the cell. However, we can specifically say that is occurring partly in the nucleus and partly in the cytoplasm. And the process of protein synthesis properly, as biologists, these terminologies are very, very vital. One of the terminology, the term known as chromosome. Remember, chromosomes, these are thread-like structures found in the nucleus of a cell. Remember, these chromosomes, they are polymers. They are made up of smaller units known as DNA. Meaning, from chromosome, you will go to a second terminology known as DNA. DNA, we are saying that is the deoxyribose nucleic acid. This is a store for genetic information. It is like a memory card having all the instructions of any process which is occurring in a body, any manufacturing, any catabolic process, any anabolic process. So it is the architecture plan of the whole biological body. So the way you look like outside, the way you look like inside, this is the commander. Therefore, these are monomers of the chromosomes. Remember, DNA is having special features compared to its sister, known as RNA. Remember, DNA, what is important to note, DNA is double-stranded. DNA, when you discover, it is made up of the nucleotides. Nucleotides, these are smaller units. Remember, nucleotide, you will find there pentose sugar, then you will find there the bases. The bases which are common in DNA, as you can look at that cursor, they are mainly adenine, represented always by A, letter A, capital A. Then we have cytosine, represented by letter C. Then we have guanine. Then thymine, which we represent it by T. So when you look at now RNA, you discover that RNA, instead of having thymine, then for it will be having what? Uracil. So we see in RNA, like messenger RNA, our adenine is going to now pair with uracil instead of pairing with what? Thymine. So take note of that difference because it is very, very vital. RNA is single-stranded. Then too, you'll discover that RNA is having uracil in sin. Now, when you are going to discover that in DNA, the bases pair up. You discover that adenine is pairing up with thymine and cytosine with guanine. So we are going to discover now that the pairing up is accomplished by hydrogen bonds which are weaker. Therefore, DNA is in the form of a radar. This explains why books will tell you it is helical, an helix. It is a spiral. It keeps on like coiling like a snake, but in between there are some bonds, as you can look at the image you have there. So DNA, another thing to note about it, it is semi-autonomous and semi-conservative. In simple terms, it can make copies of its what? Copies of itself, and so on. Now, DNA structure should be understood. We are going to understand more the structure of DNA when we look at what we call DNA replication. So another terminology which is important is what we are calling a gene. Now, the big molecule we are calling the chromosome. The chromosome. Then the monomers of the chromosome, we are calling them the DNA. Now, in the DNA, there are some portions carrying specific instructions. Now, that specific part of DNA that is carrying an instruction or a particular code for manufacturing a particular protein is what we are calling now the gene. So we can simply say that a gene is a portion of DNA carrying a particular instruction DNA that code for a polypeptide chain. Or we can even say a gene is a portion of a nucleotide that code for RNA molecule. Remember, in your genetics, you always simplify everything by saying a gene is the basic unit of inheritance. Don't forget that. Remember Mendel's biology. Garuga Mendel, the father of genetics. That man reflected a lot there. Now, other words which we need to understand properly, guys, we should clearly know what we call transcription and translation. Remember the process we are going to talk about of protein synthesis, it mainly occurs in two major phases, even though there is a third one which is minor. So we are going to look at clearly what we call transcription, then we look at what we call amino acid activation, then we look at what we call translation. The moment you understand translation, you understand transcription, translation, and amino acid activation, there you are going to say that you have got the process of protein synthesis. Forget this, that DNA can replicate, so we shall talk about even DNA replication, how DNA is making a copy of itself. So to understand properly protein synthesis, a student must understand properly what we are calling DNA structure and what we call RNA structure. Because the commands are going to begin from the DNA structure, then the command will be carried by RNA. The DNA will be transformed into RNA. Then it is the RNA to take the pollen. So we are going to be having our DNA as the engineer. Then we are going to look at RNA as our foreman at the site. Then we shall unit of inheritance or the sequence of nucleotides that are going to code for a RNA molecule, but we are going to discover the genes occur in the form of genetic codes. They occur in the form of a genetic code. And the genetic code, as it was stated by Watson and Crick, remember those ones are very good pioneers in the discovery and structure of DNA. So genetic code, as was said by Watson and Crick, is a simple relationship between the bases and the amino acids. Now we are going to see that a genetic code is the one having the bases which are going to be complementary to the amino acids which are needed in making the protein. So that relationship between the bases and amino acid that we are going to call it a genetic code. The bases which are going to be in the genetic code, they are always occur in the form of a triplet, and these bases include the adenine, guanine, thymine, and cytosine. Remember, adenine is always represented by a capital A, guanine with capital G, thymine with capital A, capital T, and cytosine, capital C. We are seeing that the four bases, as we are saying, the adenine, guanine, thymine, and cytosine. But remember, when we shall see in the messenger RNA or in the RNA, we see that part of the adenine is pairing up with what we are calling uracil. Apart from the starting bases and the ending bases, all the other bases we shall see where there is thymine, we are replacing with what we call uracil. Take note of that. In DNA we have thymine, but when you come to RNA we shall see uracil, apart from the ending and the beginning portions of the RNA molecules. So, members, I was saying that to make a polypeptide chain or a protein, we need 20 common amino acids, and these amino acids are going to be coded by 3 bases which are calling codon. Each amino acid is coded by 3 bases which are calling what? Codon. And therefore, to code for all the polypeptide chain, the all peptide chain, therefore, to code for the whole polypeptide chain, there must be what we are calling 64 codons. That one now, the simple mathematics is going to be like each codon is having three bases mainly. But remember, if you're having three bases, you're having three bases in the codon, and you have 4 bases which must pair up: the adenine, the cytosine, the guanine, and the cytosine. You'll discover that now the new mathematics is going to become the 4 times 4 times 4, and we shall end up reaching what we are calling 64. That one can easily be reflected by that table which I have shown you there. So in simple terms, for a single amino acid to be coded for, even though we are talking about one codon, but we have 3 other codons behind. In simple terms, we need 4 codons to code for a particular amino acid. So when you look at the whole peptide chain now of 20 amino acids, the mathematics is going to take us to 64. But not all the 64 are involved in forming the protein. We see that the first one is acting as a starting codon. Then the last 3 acting as the useless or the no-sense codons. So in simple terms, if you are going to go in deep biology, you will discover that the codons which are really needed are going to be 60 in number, because the one which we are talking about, the beginning one, is acting as a starting codon. Then the last 3 are acting as the no-sense or the useless codon. So understand genetic code. That a genetic code is simply a relationship between the bases and amino acids, which are going to be now used in forming polypeptide chain. Remember, the genetic code we are saying is existing as a triplet, meaning now made up of codons with 3 bases. How many codons do we need to form the whole amino acid? We are saying we need 64. We need 64 codons. Now, when you look at, for example, our cursor there, watching now the information you have on your screen, you'll discover that DNA is having its own bases. Now, DNA can make a copy of itself. The process of DNA making a copy of itself, that is what we are calling the That's what we are calling the replication. When DNA is making a copy of itself. So we are going to see that in the process of DNA making a copy of itself, the DNA helix or strands are going to unzip or unwind. This one is mainly done by an enzyme known as the DNA helicase. When the DNA opens up, it forms a replication fork, like the ending part becoming exposed. In the same way as you can open up your zip. Then from there, we are going to see that the DNA polymerase, we have what we call DNA polymerase. DNA polymerase now is going to carry what we call the daughter nucleotides. Nucleotides, these ones, remember, they contain what we call bases. And then on their skeletal plan, they have what we call a pentose sugar. In simple terms, the bases are being held by a sugar, which is a pentose sugar. And if it is a sugar in DNA, we say that is deoxyribose. If it is RNA, we say that is ribose. So the DNA brings now nucleotides, which are complementary now. You'll find that when the unzipping of the DNA, that is the opening up of the DNA. When it opens, as you can see there, it exposes the bases. When that occurs now, the DNA polymerase is carrying the daughter nucleotides and is going to align them. Remember, as is aligning them, they are going to be complementary. So if one strand of the DNA which is exposed, the template one, the one which is exposed, we are calling the template. So after the nucleotide, the daughter nucleotides are aligning themselves along the template strand. That is the old strand from the original DNA. Now the new one is one where the nucleotides are forming. As you can look at that cursor, try to follow up that cursor as we are forming our new strand. Now, duplication, you are like a photocopy. You make a duplicate. But what you are making a duplicate, the bases in the old strand must be opposite to the bases in the new one. So if they opposite to each other, this explains why I'm calling them what? Complementary. The other one is in the what? The template. So after the nucleotides arriving themselves properly, DNA polymerase does the work, joins the bonds in between the bases, and then that strand is going to be now linked to the other old strand. So you'll find that we have formed two strands of DNA, which now is the DNA molecule is going to break off and it begins its independent life. So because the old strand is kept there, so this section is why we are calling DNA is semi-conservative. Reason, the double helix is composed of both an old strand and then the new strand. The new strand from where? From replication. How have you done replication? The DNA molecule and zipping in the presence of DNA helicase, exposing the nucleotides or the bases, and the strand which is exposed, the old one, is acting as a template. Then what happens? The DNA polymerase is going to line up the nucleotides, which are complementary, opposite the other ones, which are in the old what? Old strand. Then afterwards, the other new molecule is going to break what? So members, we can generally say that the whole process of protein synthesis is mainly subdivided into 3 phases. Don't forget that. So the first one, to understand the process properly, we have a phase of what we are calling transcription. Transcription means we are going to see forming the messenger RNA. That process, our DNA is going to form our messenger RNA. It's going to become what we are calling transcription. Then we are going to see that after transcription, we are going to do what we call amino acid activation. Then after doing amino acid activation, we are going to do what we call translation. Translation, we are going to see now our plan, which has our messenger RNA being converted or being read and converted into what we call polypeptide chain. Members, remember we are saying the instruction is to make proteins. The instruction to make particular proteins are mainly contained in our DNA. Remember, the DNA is containing what we call the genes, and we said a gene is simply a continuous string of nucleotides that codes for messenger RNA. Now remember, our messenger RNA is one which is going to carry the information of the protein which you want to do what? To make. Now the gene is having a compartment in the DNA. Remember we said it is a portion of DNA. DNA is the whole thread or strand. Remember DNA we said is a monomer of chromosome. It's a smaller unit of the chromosome. But inside it again is having small compartments or portions which are calling the gene. Now a gene is located, for example, a gene is controlling information of air or an enzyme. A gene is going to be a portion beginning from what we call a promoter end and then ending with what we call terminator end. That is now our gene. Now we are going to first reflect on what we are calling transcription, and we see exactly what happens under transcription. But before we look at transcription, just remember that the genes also contain what we call the regulatory sequences. Regulatory sequences. These regulatory sequences are the one which I called the codons. Remember we said the codon is made up of 3 bases. Now those regulatory sequences or the codons also the kind of triplet form. Now remember, before we reach the point of transcription, we are changing our command of DNA instructions of DNA into a protein. But who is going to work as the middleman? The middleman is messenger RNA. So I want you to put this one to let me bring it to a local example. You are looking at our planner. Our architecture planner is the DNA. Our architecture planner is the DNA. Now who is carrying that plan made by DNA is what we are calling messenger RNA. Messenger RNA is taking where? Messenger RNA is taking it to the site. The site is our cytoplasm. And inside our cytoplasm is the engineer at the site. The engineer at the site is one we are calling the ribosomal RNA. Is one who is going to read the plan to interpret for the builders. Then the builders, we have transfer RNA is carrying now the materials, and those materials which you are going to use, like as you are carrying sand and bricks, the materials for making the protein we want are going to be what we call the amino acids. Who is going to read them and bring them? The ribosomal RNA is reading, and then the transfer RNA is carrying to the ribosomal. Now to make that full chain, which you are going to see in what we call the translation. Remember, like the transfer RNA is having what we call the anticodon, is having bases which are opposite to the codons which are in messenger RNA. Remember, messenger RNA is carrying codons, or what we call the instructions. Then the transfer RNA to carry the proteins which are going to build, as we are going to see in that video, is going to be having the anticodon. Those ones which are opposite to what we are. To convert the instructions carried by messenger RNA into a protein. Now that one we are going to call it the gene expression. Gene expression. Now we are converting the codons in the messenger RNA into a particular polypeptide chain. Now the whole process of gene expression is what we are going to refer to protein synthesis, and it is mainly divided into what we are calling transcription and translation. Amino acid activation is just a minor process under what we are calling translation. Now when you look at that video, one side you are going to see it is occurring in the nucleus. That is what we are calling transcription. Another side is occurring in the cytoplasm. That is what we are calling translation. During transcription, we are seeing that the DNA is going to act as a template for making messenger RNA. The DNA is going to act as a plan for making messenger RNA. How does it occur? We said the DNA is double-stranded. So what happens is the DNA is going to unzip. So during transcription, we see three stages occurring. We are seeing what we call the initiation, we are seeing elongation, and we are seeing termination. Now the whole process of forming messenger RNA, which we are calling transcription, is going to be initiated by RNA polymerase binding with the promoter region. The promoter region, it can either block or allow the binding. If now the RNA polymerase binds the promoter region of the gene or the portion of DNA, now we are going to see that now free nucleotides now are going to be aligned by RNA polymerase, and this one we are seeing elongation. Elongation is now free nucleotides being arranged along now the strand, the template strand of what? Of DNA. Now during termination, we see that RNA polymerase now, as it is reaching, following the template, which is now the other single strand of DNA which was exposed. Now time is going to reach when the RNA polymerase reaches the ending part, which we are calling the terminator region or terminating region of the gene. That is the point when now the RNA ceases the linking of the nucleotides, and that is when now we can say that the messenger RNA strand has been what? Has been made completely. And now it's going to the RNA polymerase is going to dissociate, and after dissociating, we are going to see our messenger RNA passing via the nuclear pore through the what? The cytoplasm to begin the process of translation. So in simple terms, transcription, what are you going to talk about? We're going to talk about binding of the RNA polymerase with the promoter site of the gene. That is the beginning part of the gene. Then from there, we are going to see what? The RNA polymerase begins arranging the nucleotides following the template of DNA. That is the unzipped DNA. This implies what? Binding of the RNA polymerase with the promoter region unzips the DNA, makes the DNA to open up, and it's such that the RNA polymerase can now move along the open single strand as is aligning what you call the new nucleotides. As is aligning the nucleotides, time reaches, because it's linking them, forming bonds in between them. Time is going to reach when it's reaching the ending part, that is of the gene. That one we are calling the terminator end of the gene, and that will be the end of the chain of messenger RNA. And we shall say that the process of transcription has been what? You look at that video, you're going to discover that the form of the messenger RNA, the messenger RNA which we have formed the members, contains different regions. The formed messenger RNA contains different regions. It contains regions known as the exons. These regions are those ones which are going to code for our protein we want. But again, is having regions which are calling the non-coding regions or the introns. Now, those introns are useless in our messenger RNA. Now, a mechanism is done to cut out all of those the non-coding regions because they are useless. They are making the strand longer, but when they are useless, we are only interested in those ones which are red, which are calling the what? The exons. So we are going to look be removed. The introns are going to be removed by a process known as intron splicing. This one is mainly done by what we call the splicome. Splicome is just a combination of the proteins and then what we call RNA. These ones are going to be chopped off. You see that the yellow ones, which are called the introns, are being chopped out. Like as you can cut out the yellow ones and leaving. Remember, the yellow ones are carrying no codes for the proteins and non-coding. We are interested in the red ones, which are carrying now the codes for the protein which we want. So we are seeing them being removed, and as they are being removed, we are going to remain with our messenger RNA which is complete, and then our messenger RNA which is complete is going to pass via those openings which are calling the nuclear power, and then to the what? The cytoplasm. Now when it reaches the cytoplasm, we are going to see it now. Now these codons, now they are the ones carrying our instructions which are talking about. And then out of those codons, we are saying there are four special codons. There are four special codons: the starter and the three useless ones, the nonsense one. The nonsense ones. In translation, the small ribosomal subunit is going to attach itself at the beginning codon or the static codon. The static codon. This is among now the special ones which are talking about at the 5' what? Region. Now, when it attaches itself, remember each amino acid which is going to be used in forming a polypeptide chain is having particular bases. Which particular bases now are going to be read by transfer RNA. So what are we going to see? Our messenger RNA is carrying instructions of the codons which are having 3 bases, but it's a lot of transfer RNA to bring. Now when you follow up our video, we are seeing now after the smaller subunit of ribosomal combining with the beginning point of messenger RNA, the beginning plan of messenger RNA, we are seeing 3 bases coming. Actually, they are showing you bases AUG. Bases AUG, those ones are from messenger RNA. Then we are seeing the transfer RNA having what we call the anticodons. The transfer RNA is having codes which are opposite to the codes which are in messenger RNA. So if you have A, now the transfer RNA is having uracil. Adenine transfer RNA is having uracil. Now when you are seeing that our messenger RNA is having uracil, the other transfer RNA is reading adenine. Then we are seeing our messenger RNA is having guanine, the other one is reading cytosine. So meaning the anticodons of transfer RNA, you are going to look at our transfer RNA. By that video, you can see that is one coming and is being fixed into the messenger RNA. But remember, who is anchoring, who is holding down? That is the ribosomal small subunit. Now the process is going to continue like that. As ribosomal continues, now we are going to see the bigger ribosomal subunit is going to fix there. To now to wrap up that beginning point, as the process is going to continue going the other side. Now in simple terms, the process of translation, which is now the final process of giving us our protein which we want, which now we started with from the commands from DNA, is mainly involving the following. We are going to see the binding of messenger RNA into the ribosome. Then we are going to see the amino acid activation and attachment to transfer RNA. Then we are going to see polypeptide chain initiation, then chain elongation, then chain termination, and then we shall look at the fate of messenger RNA. What happened to messenger RNA after doing all its work? So far we have seen the smaller unit of ribosomal RNA attaching itself to the messenger RNA carrying the plan. But remember, the messenger RNA, as you are seeing different colors illustrated on that model, they are different colors, but they are occurring 3. The other smaller unit of ribosome is attaching at the beginning, and that is at the 5 end. That is where we have the starting codon. Then at the three end, there we are going to have what we call the nonsense code, where the ending of the polypeptide is going to be. Now you are seeing transferrin has carried the protein or amino acid. Look at the top there. The purple ball there at the top of transferrin A. That one is what we are calling now our amino acid. Now with other transferrin A, remember you have millions and millions. Others are going to come also. As they are doing what this one has done, we are going to say chain growing slowly as they are adjusting on the left hand side. They are going to adjust on the left hand side. Adjust like as you are moving in a line, as you are moving, replacing your friend, and so on. So that first transferrin A, which has combined with the beginning codon of the messenger RNA, is going to transfer RNA, which has combined with the beginning codon of the messenger RNA, is going to work as the initiator transfer what? RNA. And now we are going to see the big unit of ribosome coming there to fix, to form what we call translation complex. Translation complex, you have combined transfer RNA, that the initial transfer RNA, then with the beginning base of the messenger RNA, then with the first amino acid. You can see you are forming a skeletal plan now, where other amino acids are going to be arranged as the chain is becoming what? Becoming continuous. And that is what we are going to call initiation phase, initiation process, the beginning point. Now with the second stage is going to become what we call elongation. During elongation, we are going to see more amino acids being brought. More amino acids are going to be brought by different transfer RNAs. And as transfer RNAs are bringing different amino acids, we are going to see a chain growing slowly. Follow up the purple. The purple is going to grow slowly. The purple balls are going to be added. But who is adding? The transfer RNA. How does transfer RNA add? Transfer RNA add by having bases which are opposite to the bases in the messenger RNA. We are calling them the anticodons. They are anticodons because those ones on messenger RNA, we are calling them the what? The codon. So the chain will continue on elongating. Now remember, as we are adding more amino acids being brought by transfer RNA, you can see that arrangement, that there is some space which is being created by the next transfer RNA as the chain is extending to the right hand side. The transfer RNAs which have been used now for them, they are departing via the other side of where we started from. Now this implies that amino acids must be now eroded stronger. That chain which is up, made up of blue and purple balls and the brown ones, must be now eroded. The amino acids must be joined, and they are going to be joined by what we call peptide bonds. So we see some peptide bonds being what? Being formed. Now the process is going to continue as the chain is elongating until when now we are going to see we are reaching almost the ending point. Try to follow up you will see that when you count from the right hand side from the three end, you will see that now this 1, 2, there are three now. There are three codons which are useless. Three codons which are useless on your right hand side. Now as the ribosomal RNA is sliding and it almost reaches the three end, where we are seeing in near to what we call the nonsense codes, we are seeing that a release factor now is going to be at that ending point. And now we are going to see that the small unit of ribosome and then the big unit of ribosome are going to dissociate, and then the initiator transfer and dissociate. The whole complex dissociate. Remember at the beginning of the chain they were combining. Now we are seeing them separating in the same way like you are initiating as the start. That is the point where they are doing some modifications. So now remember, as to wrap up translation. One, we are seeing messenger RNA is going to bind with smaller unity ribosome RNA. Then we are seeing initial transfer RNA combining. When it combines, it's going to now combine with it, reading the starting codon. Then we are seeing a big unit of ribosome combining to form now the initiator complex. Now we are seeing now the initiator complex sliding on towards the right hand side as different transfer RNAs are bringing what we call amino acids, which are being joined by peptide chain, joined by peptide initiator complex,
slides to the right hand side until when now it reaches the ending side, the 3 end. Remember the chain is being formed from the 5 end starter codons, then the ending side of the chain, that is the 3 end. Now what are we going to see there? We're going to see now a release factor. What you call now releasing polypeptide chain is going to occur. How are they releasing? Now the other ones are going to separate the smaller unit ribosome, the big unit ribosome, and then this initial transfer in the separates, and the chain is released, and the chain can be modified either to primary structure, secondary structure, quaternary structure, based on what we do, based on what we want to make. If you are going to make hair, the chain is modified into keratin, what we want. If you want to make a nail, the same. Enzymes, some modifications are done in the Golgi apparatus. You go back to cell biology. That one will be clearly understood.